objectives of the day are students will be able to critically analyze the history of chromosomal theory with reference to Corin's work. Introduction. In 1865, Gregor John Mendel, who was a famous geneticist, used the term hereditary factors. At that time, the location of these hereditary factors was unknown. No one was aware of even the cellular structures or the organelles. With advancement in molecular genetics, we can define the gene or a hereditary factor and we can describe its location. What is a gene? A gene, by definition, is a small part of the DNA or the deoxyribonucleic acid that is a sequence of nucleotides. It is responsible for synthesis of a specific protein by the process of transcription and translation. Or simply, in a single sentence, you can say that gene is a small part of the DNA that is responsible for the synthesis of a specific protein. Where is it found? When we study the chemical structure of the chromosome, we find that chemically chromosome is made up of DNA and proteins. The proteins are the histone proteins, where DNA is the deoxyribonucleic acid. So the small parts of this DNA, which are having the information for the synthesis of a specific protein, these small parts are termed as the genes. So today we say that the location of genes is in the chromosome. Genes are found in the chromosomes. Now we start the history of formulation of chromosomal theory of inheritance. And the foundation for formulation of a testable research-based theory of heredity was laid by the work of Gregor Mendel and Darwin. Mendel was a geneticist and Darwin was the naturalist who presented the theory of evolution by means of nature. What is chromosomal theory of inheritance? By definition, the idea that genes, the units of inheritance, are found in the chromosomes. So chromosomes act as carriers of heredity. This is the definition of chromosomal theory of inheritance, that the units of inheritance are found inside the chromosome. Now first is the role of or the contribution of Walter Fleming. In fact, the chromosomes were discovered by German embryologist Walter Fleming in 1882 while observing the dividing salamander larvae. Voltaire proposed the term chromosomes, where chrom means color and soma means body. Karl Correns. In 1900, the German geneticist showed the idea of relationship between genes and chromosomes, but was unable to prove it. In 1902, Walter Sutton, an American graduation student, and Theodor Boveri, a German biologist, recognized independently that behavior of Mendel's factors is parallel to the behavior of chromosomes. Objections were raised to the Sutton's theory. If we accept that Mendelian traits are determined by the factors located on the chromosomes, and if the genes are segregated due to segregation of chromosomes and independent assortment of genes is reflected by the independent assortment of chromosomes in meiosis, why is it that number of genes that assort independently is often greater than number of chromosomes pairs in the organism? T.H. Morgan's contribution. Discovery of phenomenon of linkage in Morgan's famous eye color experiments in Drosophila melanogaster, removed the objection from these essence theory. So now the definition of chromosomes. Chromosomes are the physical carriers of hereditary information. Morphology of the chromosome. In a cell which is not dividing, we can see the chromosomes in the form of very fine network, and that is termed as the chromatin network, and that happens during interphase. When the cell division starts, mitosis starts or meiosis starts, we find that the chromatin network condenses, it becomes short and thick, and it becomes visible in the form of structures known as chromosomes. Now, the structure of chromosome. The chromosome consists of two thread-like structures termed as the chromatids. The chromatids are joined at the central point termed as the primary constriction or the centromere. A primary constriction or the centromere is the area where we find spindle fibers attached during cell division. Then another constricted area can be found along the length of the, or it can be seen along the length of the chromosome. This is termed as the secondary constriction or nuclear organizer. This is in fact the point where new nuclei are formed during interphase. Now, if this secondary structure is near to one end of the chromosome, the part of the chromosome beyond this secondary constriction, knob-like structure, and this knob-like structure is termed as it consists of the useless DNA. Now, what are the ends of the chromosome known as? The term telomere is given to the terminal ends of the chromosomes. Now, what is the importance of this telomere? The telomeres are responsible for keeping the chromosomes unattached. Different types of chromosomes on the basis of the position of centromere, starting with metacentric. Metacentric, where the chromosomes, these are the chromosomes where the location of the centromere is in the middle, and it results in the formation of the arms of the chromosomes which are almost equal in length. Next is submetacentric. Here we see that in this diagram that centromere is slightly displaced or moved from the middle point or central point. And such in this way or this type of location of centromere leads to the formation of unequal arms. Acrocentric, almost towards the end, where you can see that the centromere is present near the end, and it results in the formation of a very short arm of the chromosome, and the other arm is long. And the last one is telocentric. Telomere is known as the end, so telocentric, where the centromere lies at one end of the chromosomes. Thanks.